Welcome everyone. My name is Jen Lynch and I am the Marketing Director for Rigaku Analytical Devices. On behalf of all of Rigaku, we want to wish you and your families health and safety during this global pandemic. Before I introduce our presenter, I want to let you know this webinar is being recorded and you're all currently muted. We welcome questions to be submitted via the Q&A section throughout the webinar, or you may hold them until the end when we unmute. Also, throughout the webinar, we will ask a few poll questions and invite you to participate. Now let's get started. Jill Carrero is our Global Business Development Manager and has years of experience in handheld analytical solutions. Today, she is going to present using the Rigaku Progeny 1064 Raman as the most versatile solution for your raw material ID needs. Jill?
Good morning, everyone. I hope you're doing well. It's a strange time in the world, but I'm glad that it gives us, a lot of us, this opportunity to connect in a way that maybe our busy day-to-day lives don't often allow us. I certainly hope in your personal lives, you're also taking the opportunities to smell your family roses, so to speak, and enjoy some personal time. That said, I very much appreciate you taking some time this morning to join us in this webinar. It's the first of three. If you've joined us through our Rigaku page, you may have noticed a number of other Rigaku webinars. Of course, we are a global company offering a number of technologies, XRF and XRD technologies, as well as handheld technologies today. We're going to have a couple of webinars specific to Handheld Raman following from this one. And so if you're here to join us today to talk about Raman, great, but know that this is the first of three, and we do hope that you can join us going forward as well if you have other questions. So, Progeny, a handheld solution for raw materials ID. And this is, again, just a brief solution summary. I want to start with asking you all a question. Specifically through our polling opportunity that we have here on Zoom. We want to test this out, so please stick with us. Jen mentioned to you that I've been working in Handheld Raman for some years, and I do look forward to connecting with you about your experiences in this webinar. And starting with this question, are you familiar with Handheld Raman? If you could take a moment to answer this question, I'd really appreciate it. Wonderful. It's very interesting. Okay, so about 3/4 of you, it looks like so far, are very familiar with Handheld Raman. Great. That's what I would have probably anticipated, to tell you the truth. So let's start with what I find to be a little background on Raman, a background on Rigaku. Forgive me, I've got to clear out some of these windows so I can actually see my own window. So Rigaku, we are a Japanese company. We were founded in 1951, but Rigaku Analytical Devices specifically, we are a handheld group based in Massachusetts, just outside of Boston. We have a 15,000 and growing square foot facility. We are a 9001 certified facility that includes manufacturing capabilities. So we make all of our tools in Massachusetts. They are CE compliant and MIL standard certified. We make handheld Raman, as mentioned before, but we also make laser-induced breakdown spectroscopy instruments. So those of you who are familiar with OEM or OES, it's kind of like OES with a laser. We are very much focused on handheld technologies. There are about 45 of us under that roof there. It's not currently snow covered, and there aren't any of us in that building, of course. But we are all focused on handheld technologies. There are chemometricians, there are validation groups, there are engineers, and there are folks like me that are working specifically with customers to achieve your own personal goals in your science and manufacturing quality goals. We should mention that I have a couple of folks on the line here today with us as well from our team, Jason Walker, who leads our customer service group, and Suzanne Schreier, Dr. Suzanne Schreier, I should say, who leads our applications group. And they'll be available to answer any questions that you pose through our chat as we go through this presentation today. So what is Raman? Raman is really a 1 in a million event. If we talk about what Raman is, it's all about a very, very specific scattering of light. If you point white light at a surface, 999,999 of the photons that hit the surface of that surface will scatter back completely elastically at the same wavelengths of light. But 1 in a million photons will exhibit what's called Raman scattering instead of that Rayleigh scattering. And Raman scattering is when a shift in that wavelength of light occurs. And so back in 1928, C.V. Raman discovered this. And over many, many decades, and then what became a century later, we have developed technologies now in the field of Raman technology that allow instruments that used to be the size of, say, a large bench or a large lab test desktop to be miniaturized to the size of a handheld instrument. This was very, very much a success story for different industries about a decade ago, including safety and security industry, that utilized this miniaturization of Raman spectroscopy for a lot of advantages. Specifically, when we're looking at Raman scattering, and if you look at here on the left side of my screen, you'll see that what happens is we use, instead of white light, we point a single wavelength of light, a laser. In the case of Progeny, it's a 1064 nanometer laser at the sample. And then what we see is Raman scattering of that single wavelength of light. And we measure the Raman scattering that happens as a fingerprint that's specific to that molecule or element. And when we can do that, we get a very, very unique advantage through Raman spectroscopy to give an idea of what that material or element is. So I'd like to ask you another question. Jen? I'm looking for the polling. So the next question. So if you use a handheld technology at your site, which one do you use? Do you use your Raman? Maybe Progeny? Maybe another Raman? This is very interesting for us. Thank you very much for taking the time to answer these polling questions. Okay. So welcome to those of you who use Progeny. Thank you very much for returning to us. And if you're familiar with another handheld Raman, we definitely hope that this gives you some insight today to what makes Progeny different. But looking at this slide here before us, you know, to answer or to share these results with you, I'd like to share these results with you. So the majority of you, well, about a third of you, if you see here, are using Progeny. Many of you are using another type of Raman. And then some of you don't do any testing at all. So there's something to learn here today too for you. So I'm very excited to have you. Thank you again. When we look at vibrational spectroscopy and compare the different techniques that are available, you know, we can really understand very clearly some of the advantages of the different technologies. And when we look at Raman, near infrared, and FTIR specifically, there are different advantages to each technology that stand out. I like to go right to specificity, and specificity is really the ability of the technology to tell one material from another. And when we're talking about specificity, the ideal technologies are Raman and FTIR. And if you look at the bottom right of the slide here, you'll see why. And it's because you see those nice, sharp, clear peaks. And that makes it really easy for an instrument like Raman or FTIR to tell the difference between one material and another without a great deal of method development or validation. Now, near infrared offers less specificity, but you know, there are other advantages that near infrared can offer, like being able to tell the difference between particle size. There may be an application that you want to be able to tell the difference between particle size. So near infrared would be an ideal solution for that. When we're talking about sample preparation though, that's really going to be such an important aspect to what we do in the realm of pharmaceutical raw materials analysis, right? We want to be able to do our sample preparation with as little contact as possible to our product. And so Raman allows that better than any other technology. So those two advantages combined make Raman a really ideal solution for RMID. You can avoid the sampling risk of contamination, forgive me. It's a non-contact analysis, right? So you get to test through your transparent and translucent containers, and you get those measurements in seconds. It's really a built-for-purpose solution with incorporated analytics. So I'm gonna reach over here. You know, Progeny, it's a solution. It's a solution in a package. It's made to be the chemometrician and your quality analyst in the package. It gives you the answers that are necessary to do your raw materials analysis without a great deal of at point of use analysis capability. It's pass and fail very quickly. It's very easy for the non-scientist to operate. It's industry accepted for raw material ID. So US International Pharmacopoeia, we'll touch on a little bit more on that later on, but it's really as analytically effective as conventional methods. And about 10 years ago, when I was talking with pharmaceutical companies about using handheld Raman instead of their benchtop and near infrared, this was really a challenge because nobody was using handheld Raman in the pharmaceutical industry. But nowadays, we understand the great advantages. We're written into multiple pharmacopoeia, specifically referencing handheld Raman. So certainly, it's a great go-to tool for this purpose. And really, it's accessible. You can increase your inspection rates. So if you have a challenge to do the work that you need to do and do it more quickly with less bodies, get more awareness to your product and reach more markets, handheld Raman is a great way to make a leap to do that. It changes the workflow. There's no doubt about it. And I know a lot of folks out there, Jeff Weaver, I have you in mind. I know you're on the line, so I have you in mind when I look at this slide because it's easier said than done, right? But we are here to help you with this. But when we look at how the workflow has changed, really you can look at that red chunk of graphic there to think about what's removed from the process when you go from a traditional workflow over to a handheld Raman workflow. It really allows you to make a jump, to remove the labeling, to remove the transfer of material to a sampling suite, the gowning and cleaning process that goes along with that, quarantine times. Oh, I said the word. Can't believe I said the word. It has a whole new meaning in all of our lives, doesn't it? It really streamlines the process and reduces the amount of time necessary for the turnaround for the ID process. You're still going to have to go through some compendial tests, but the process of taking product and opening containers is really minimized when you're using Raman for handheld raw material ID. And when you're looking at it with Progeny, this is what it really feels like to the user. These are the 10 screens that a Progeny user sees when they're going through the process of approving a raw material when it comes in for inspection. The first step would be to choose the scanning process. They would select the application. So specifically, that would be the raw materials application, or maybe your company wants to break it up into the solvents and the powder raw materials that you have. They're going to have directive to pick the correct material, and that can be done using the barcode scanner that's on the instrument. And then they're going to point the front of the instrument at the sample, whether that's through an amber bottle or through the liner of a bag, the liner of a drum, or through a number of other transparent or translucent containers, and they're going to tell the instrument to start the scanning process. And through the preset scanning parameters that have been developed through the quality analyst and development process on your Progeny when you set up your instrument and develop your methodologies on the instrument will go through and compare the Raman spectrum that is gathered from the scan to what's stored inside as the reference spectrum on your instrument, and uses the preset criteria in the algorithm to determine whether or not it is a pass or fail. And so specifically, we can talk about some of the criteria that is available to you, but I want you to take a look at the bottom right screenshot there. It gives you a great understanding of what the analyst sees as the final screen. That's a pass, so meaning that the sample that was received was the correct sample. It matches what was expected to be received. And you can see here that for this test, it was a 0.99 correlation coefficient match. And you can see the overlay of the spectra here. And there's a lot more to be learned just from this single screen by swiping and looking at more information that can be revealed on Progeny. And we'll touch on that a little bit more here going forward. So really, to give you an overview of the complete package, it's really fit for purpose. It's a very flexible user interface here. And so I just touched on that just a bit. There's the availability to swipe on the screens. So I'm trying to show you here. When we have information available on the screen, we can swipe left and right. We can zoom in to make more information available to us. There's an integrated digital camera and barcode reader. We have vial holders. We have tablet and bag holders. It's a completely 21 CFR compliant solution. And we have a sync software package that allows you to integrate fully into your LIMS package, to allow a completely offhand data transfer from the end user into your secure sync location, and then parsing out into any LIMS package that your company uses. We provide, of course, full IQ/OQ and SOP documentation and full customer support for 2 years. So really, to talk about the details of why Progeny is so ideal for raw materials identification, it's because 1064 allows us to see much less fluorescence. And fluorescence is just a natural spectral occurrence which interferes with the Raman signal. If you look at the bottom right here, the red spectrum is really what fluorescence feels like in a Raman spectrum. And that's what we often see when we're using, say, a 785 nanometer based instrument versus a 1064 nanometer based instrument in Raman. When we're looking at the spectrum, the red line spectrum there, that makes it almost impossible when we're looking at an example like that to take any Raman analysis, any real spectral information from that spectrum. Now, it doesn't always look like that. Lots of 785 instruments provide lots of great data. But the thing is, and I could tell you firsthand experience because I worked with some of those instruments for many, many years, is that even when you get really great spectral data with a 785 instrument and you don't see a big hump of fluorescence like that, you still feel the fluorescence. The fluorescence is still present in the spectral data. It still must be processed through, and it still slows down the time of analysis. So scans might be limited, their scan time. You may not get all the information that you need. And some of those materials that you work with, it just might be absolutely impossible to get any information from. Progeny, based on 1064 nanometer laser, is just much less sensitive to fluorescence issues. It's nothing special about Progeny. It's nothing special about Rigaku. It's 1064 that makes that special. And I always like to talk to folks about, you know, really that if 1064 was available as a solution, as a smaller miniaturized solution, earlier, the folks that had made the first Ramans probably would have chosen 1064 because it's a better solution for raw materials identification because it experiences less fluorescence. So it extends the number of materials that you can test, and it just makes all of your testing much, much faster. Samples that used to take 30 seconds just take about 5 seconds. It's just a fact of the matter. What this also really adds to the mix is the ability to test many, many materials that you can't with a 785 instrument. So some of those problematic materials that are easily detectable with a Progeny are like xanthan gum. Oh, I see a spelling error there, forgive me. The polysorbates, cell culture media, so many different cellulose products and excipients that experience slower scan times or just really difficult to analyze materials are very easy with the 1064 instrument. And then on top of that, everything that is easy with a 785 instrument is also very easy with the 1064 instrument. When you have 785, honestly, the advantage is that usually it's a higher laser power. And so when you're talking about very, 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 very small amounts of product in a very large mixture, sometimes 785 is better. But otherwise, 1064 offers much, much more advantages across the board. Here's a short list. And it really is a short list of, and non-inclusive for that matter, of the materials that are much better for 1064. Anything that's colored on your site is going to work better with 1064 instrument. And so that's something to consider. Anything that's a long chain polycarbon, that's going to work better with a 1064. Natural materials, things like that. If you have materials that you can't test with your 785 instrument, if you're one of those folks, that 25% or so folks out there that are using a 785 instrument, and you want to use you want to see what a 1064 looks like, please contact us. We'd be happy to test some of those materials. So looking at just what the Progeny feels like when you're using the instrument, the experience of using the instrument, it's a great tool because it's got a huge touch screen, and you also have the buttons to work with. It's got a barcode scanner and camera. So what this allows you to do is take pictures of your compromised sample. I can't tell you how many times there's my rooster going, how many times I've had experiences with customers where they've told me that they've opened up a drum and had an open poly bag. Now there's an ability to detect or to capture an image of that and have it right with the report, incorporated with your onboard report on the instrument that can be uploaded to your server later on. There's an adjustable focus nose cone that allows you to control the depth of laser penetration. So there's been some questions that have popped up around translucent and transparent containers. The rule of thumb is if you can see the product, let's give Progeny a try. If it's a completely transparent container, there's absolutely no doubt in my mind that you're going to have some success reaching your material with Progeny, with the laser and Progeny. Now that said, the materials that are like amber bottles and thick translucent containers, they are made to block light, and laser is light. So we have to do a lot of validation and understanding around those type of containers to see if Progeny can be used with them. But that said, when we're using Progeny, we have an adjustable focus nose cone that allows us to look through all sorts of different containers. And we have some really great videos on our website now that show you how we can look through different types of containers, specifically 6 different types of containers with the Progeny, with a single method, and how it can be used for different containers without any challenge. It's also, to be very clear, a sealed system. So this can be, it's an anodized aluminum system, so this can be completely washed down. It's a real advantage to any rubberized system that you might have on site for sanitization purposes. To look at the software, it really is a huge, huge advantage when you're talking about Progeny to alternative solutions that are out there today. It's a really intuitive solution. We offer a touch screen that allows a super fast learning curve. Swiping is how we all function in our day-to-day, right? When we use our phone, we swipe left and right. Well, that's exactly the experience that Progeny allows you to. When you're on a screen, and if you want more information, a simple swipe left or right will often reveal the spectrum, will reveal the specific information about the scanning parameters used to collect the information, all sorts of good things. Also available in Progeny software is the ability to fully control all of your analysis parameters. So this is something that can't be done on other software packages like it can be done on Progeny. And specifically, it's done on the Progeny. So the algorithm allows you full control of whether it's a HQI or wavelet-based algorithm. You get to pass the correlation or set the correlation coefficient pass and fail values, the power, the exposure times, scanning ranges and search criteria, and the libraries and how the libraries are referenced. There is just such a greater level of control and accessibility in Progeny than with other software packages. And the average scientist, where, you know, the tool is made to be used in the warehouse. The tool is made to be used by the folks that are receiving the product and doing the pass-fail analysis and sending it through to get manufacturing going. But when we're talking about our day-to-day life and how, you know, we use the tool as scientists, we want much, much more out of it. And that control of the analysis parameters is one of those things that I know folks that use Progeny really value in the solution. So data management, you know, data management is God in our world, and really the data backup process is really an excellent one. We have a sync process that allows you to connect the instrument at the end of a workday or shift, allows a streamlined process of backing up the data into a secure location that has been dictated by you know, your IT group and your internal, you know, SOPs. Everything is fully traceable. There's a GMP audit trail available. We can obviously answer many more details on that. And specifically, we have an upcoming webinar on this topic that if you're interested in understanding how data is managed and the integrity of data with Progeny in your raw materials process, you can join us on that webinar at the beginning of May. We offer Wi-Fi on Progeny, which is really an excellent solution. Of course, you can connect via USB by the bottom of the instrument here, but we also offer Wi-Fi connectivity. And it really is a fully compliant ready solution. You know, we have taken into account the multiple pharmacopoeia in the world, ALCOA principles, 21 CFR guidelines, to give you a solution that really allows you to feel confident in how you're using the tool to both acquire your data, but also manage the data on the outbound. So why Progeny and Rigaku, to end it here, right? Well, we feel that you should consider us as a solution. And I speak from really a place of understanding, because I spoke with so many customers for many years that wanted the things in a handheld solution that Progeny offers. So it's really the most capable solution for handheld Raman, because it's 1064 and because it is what it is, because of the user interface, because of the way that it works, because of the workflow, because of the way you can move through and understand more about your data, both when you're acquiring that data and then after the point. It's also a very versatile solution. You know, the ability to walk up to two polymorphs and understand the difference between those two polymorphs as a scientist without going back to your computer and downloading data, to overlay two spectra very quickly and zoom into a very tiny peak in those two spectra and understand those differences. That's valuable, and that's possible with Progeny. And finally, why Rigaku? Well, because we really care. We are a hardworking and very dedicated team. we are 100% dedicated to the purposes of Handheld Raman. The 4 people that are here with you on the line, we are 100% dedicated to the goals that you have as a pharmaceutical quality expert, whether that's implementing the technology or 2 years down the line when you have 100% turnover and you need to retrain folks. We are consistently here for you in that goal. So with that, thank you very much for joining us. I think we went a little bit over and you'll have to forgive me. We'll address some of your chat questions now. But that said, we will also make a recorded version of this webinar available to you, of course. We also want to invite you to join these 2 upcoming webinars. And so, oh, forgive me, I've got the dates on these webinars incorrect. So the first webinar there that we have coming up after this one is April 23rd. So we would like you to join us for that webinar. It's data integrity within materials analysis with the Rigaku Progeny 1064 Raman analyzer. If you visit the website here, you'll be able to register for these webinars. We'll also have another webinar on specifically additional applications with Progeny and the ability to use Progeny and how to dig into some of those screens that I showed you or I mentioned to you to learn more about how to use Progeny in some of these more interesting applications like cell culture media and polysorbates and polymorph analysis and things like that. And then finally, if you know, in these days and age, we understand these days and age. I've been talking for too long. In this time, we understand that, you know, a lot of you are working from home. Thank you for joining us from home if that's the case. But we are available going from here to offer you any other virtual type of support that you'd like, whether that's through a private webinar for your organization. Please reach out to us and contact us for that. And my email is here if you'd like me to answer any questions. Please reach out to me. I'd be happy to connect with you personally. Thank you very much again for joining us, and have a wonderful day, and we hope to see you very soon. Thank you very much.